Hello and welcome to the lab and to the first in a series of videos about blood groups and transfusion medicine in the laboratory. Now, as it's the first one, before we start talking about blood groups themselves, it's probably good for us to talk a little bit first about antibodies and antigens and what those are. Antibodies and antigens. So we won't go too in depth, it's not an immunology lecture, but just a few concepts that are relevant to what we need to know about transfusion. So first of all, what is an antibody? Well, an antibody is, first of all, it's a protein, and as we know, proteins are long chains of amino acids. So, and in the case of an antibody, it's actually 4 chains in this sort of structure. 1, 2, 3, and 4. So it's like a Y-shaped protein there, and they're all joined together in between. So these antibodies have a long stem like this, and then the 2 arms of the Y. And then at the end of these arms, these bits at the end here are what we might call the hypervariable end. And it's important that there are 2 of them on the antibody molecule. And these hypervariable ends are very interesting. They come in all sorts of different arrangements in the protein structure. The rest of it stays pretty much the same, but these hypervariable ends, as the name suggests, can change a lot. They can have different proteins that go this way, or different proteins that have another structure, so that they're all unique. And there's millions of different kinds of antibodies with different ends. So the purpose of the antibody, and with its hypervariable end, is that it's designed to be able to recognize different antigens. So what is an antigen? Well, antigen, as the name says, it's an antibody generator. So it's something that will stimulate the production of an antibody. So it might be, for example, something like if you've got a germ. Let's make a little germ here. Here's a nasty germ. He's going to make us sick. And he may have, say it's a bacteria, there may be different proteins on the surface of the bacteria. So let's make them, say, square-shaped like this. And he might have a range of different ones. He might have some triangle-shaped ones as well, but we're just using this shape as a representation for the different kinds of molecules that may be on the surface of a bacteria, or whatever the case may be. So an antibody that recognizes the square-shaped one, for example, will bind, one of the arms will bind to that antigen, and will be able to conform to it, whether it's physically or sometimes with the difference in the electric charges, but it will be able to have some affinity for that antigen. And those antibodies can bind on in various ways. So the other term that we sometimes use for antibodies is an immunoglobulin, but which we might just shorten to Ig. Ig is an immunoglobulin. And there are probably, I guess there are 5 different types of antibodies, but the 2 that we're most concerned about is IgM and IgG. And they have slightly different structures. So the IgM molecule is actually 5 antibodies in this sort of star-shaped arrangement. And they're all joined together. And the IgG is generally just on its own like that. So 2 different kinds of antibodies which we'll probably use the most or look at the most when we're talking about blood groups. So the other important thing about antibodies, as they're produced in the body, and as you develop from a baby and start to grow, the antibodies are designed to respond only to foreign antigens. Obviously, your whole body is full of cells and different things floating around in your blood and all sorts of different kinds of tissues. And so the body needs to be trained to eliminate any antibodies that would recognize your own tissues and your own cells and your own proteins. So those are all eliminated, and the only ones that are left or that the body has the potential to produce is those that will respond to anything that's not you, all the foreign antigens that may be out there. So we must know that our antibodies are designed not to respond to ourselves, but to those things that are in the environment. So, for example, let's talk about how this might work. So let's draw you. Person here. There we go. It's my great artistic skills. So there's you. And let's say that I've got a germ. So I've got the measles, and I cough on you, and this measles virus, as it were, gets into your system. So there he is in there. Nasty little fella. So your body will first of all get sick. He does his work and makes you feel unwell and give you the disease. And in the process, your body has never seen this before. It'll start to make some kind of response. So the first response that the body usually makes is the IgM response. So it'll start to make some IgM antibodies, these little star-shaped ones, and it'll also, in the process, the body will attack this and start to eliminate it. And so you've been not feeling well while you've had that disease. So your body eliminates the virus, clears it from your system, produces this IgM, and then hopefully you're feeling better then after that, and the virus is gone. But what you're left with after you're feeling better is the memory, and that's where the IgM, the M comes from, is it's the memory function. So the IgM will remain, and certain cells in your body that will be trained to respond to that measles virus when it comes along next time. So if you're unlucky enough to get coughed on again, and along comes this virus, well, this time you won't get sick, because as soon as this virus enters your system, your body will respond using this memory immunoglobulins and also the cells that have been trained, and it will instantly start producing IgG, and it'll produce lots and lots and lots of IgE, and the cells in the body will react very quickly to eliminate this bacteria or this virus from your system before it has a chance to make you sick. So you remain happy and well, and you also have this production of IgG in your body. So the important thing to remember is this is the first response, and this is what we might call a passive. It's a slow and rather passive response, but it's there to create certain memory. And then the second response on the second exposure is this IgG, which is a very fast and aggressive response to the antigen, to the exposure to the antigen. So what does this have to do with blood groups? Well, when we're talking about immunology, we talk about red blood cells when we're talking about hematoimmunology, and so our antigen in the case of transfusion science is whatever the proteins are on the surface of a red blood cell. That looks a bit like a donut. We might just red blood cells are sort of almost a donut, but they've still got a bit in the middle. So a red blood cell has a number of different proteins or carbohydrates or lipids on the surface of its cell, and they probably have different functions, but we might give them different names. And so let's say that and the ones that are on my red cells may differ from the ones that are on yours, as we're genetically different. So let's say that you've got blood in your system, and you've got different things on the surface of your cells. Let's say you've got the C antigen, and let's say that you had an accident, you've had a car accident or something, you've lost some blood, and you're not very well, and you need a blood transfusion. We need some more blood into your system. So I put my hand up and I say, well, I've got some blood, but as it happens, the surface of my cells has the E antigen. So I might transfuse those into your into your body, and here's the E on the cells now in your body. And your body says, hang on a minute, I've never seen this before. This is something foreign and something different, and I don't think I like that very much. I'm trained to respond to and eliminate anything that I don't recognize as myself. So your body will start to produce the IgM antibodies, and against the E. So what we would call anti-E. You don't produce anti-C because that's you, and your body doesn't make antibodies against things that it recognizes as being part of you. So it would recognize this as being foreign and start to produce anti-E. And as I said, that's a fairly slow and passive response, and the blood will remain and it'll do its job, and it might over time get eliminated, but it's done its work, and you are left with this memory of IgM that's going to be able to respond to E cells. So now comes the important part, is if you were, say, to be unlucky enough to have another accident and lose a lot of blood, and you need another blood transfusion, and luckily for you, I'm there ready to help, and I put my hand up and say, oh, well, I'll give you a blood transfusion. So here it comes again. Here comes my cells with the E antigen on there. So once again in your system, and just like with the virus, the body says, hang on, I'm ready for you now. You can't take me by surprise. You did that last time, you got me, yeah, fair game, but now I'm ready for you. Here's this E, I've seen that before. I've got my preparation already. So you will start to produce IgG, anti-E. This one was IgM, but the second response, IgG. And you'll produce lots of it, lots and lots and lots. In fact, enough to straight away knock off all those red cells, which when you need them, it's not very helpful. You want them to be there to do your job, and in fact, knocking them off not only means that they're not there to do their job, but it also means that whatever was inside them is now in your body and contaminating your system, and it can make you very sick. So that's the general system with blood groups and with antigens on the surface of the red blood cells. So we call them red blood cell antigens, and they're the cause of, or the reason for all our blood groups, as well as the body's response to those antigens. So I hope that makes a little bit of sense for you, and we can understand a little bit about how antibodies and antigens work. And then in the next lecture we can start to talk a little bit more about how we detect these antigens and these antibodies in our body.